My name is Jack Castle, and I will be doing a presentation on transcranial direct current stimulation and its effects on major depressive disorder. Now, firstly, we have to understand what TDCS is, and what it is a procedure that allows modulation, facilitation, or suppression of neural activity, and it also is highlighted to strengthen the working memory within the prefrontal cortex and improve learning within the hippocampus via neuronal networks and chemical reactions. Now, transcranial direct current stimulation relies heavily on electrodes being placed on the scalp of the patient's head, and what happens is one of the electrodes is called an anode and is the electrode position, and the other one is the reference electrode, which is known as cathode. Now, the anode would be placed somewhere around FP1, F7, or F3, whereas the cathode will be placed somewhere around the FP2, the F4, or the F8. And the reason for that is to develop a circuit that allows for each other to produce depolarization or hyperpolarization. Now, depolarization works with the anode and develops excitability within neuronal networks, and the cathode is responsible for inhibitory reactions within the cortex. Now, transcranial direct current stimulation is not limited to the dorsolateral prefrontal cortex. However, it has been widely associated with the working memory, task switching, inhibition, and other cognitive procedures that is undertaken. With this, there is a noticeable reduction within the ability to use this area within people suffering from major depressive disorder, and there also has been a notable imbalance of neuronal activity within the left and right dorsolateral prefrontal cortex, which seems to be improved after stimulation via TDCS. Now, physiological responses and mechanisms of action, we have the NMDA receptors and AMPA receptors. Now, these are both responsible for the influx of sodium or calcium, which results in depolarization, allowing for spontaneous firing of the neuron activity. Now, this also, due to the influx of calcium and sodium, we also have the ability to produce long-term potentiation or depression. Now, this is determined on the modulation of sodium and calcium. If there is little calcium and sodium, it will produce long-term depression. If there is moderate calcium and sodium, then nothing will really change. However, after an excessive amount of sodium and calcium, will then see signs of long-term potentiation. You're probably wondering how the ion channels are able to influx sodium or calcium within the membrane to change the resting potential. Well, this is how it's able to do this. After stimulation, the reduction of GABA transmission is present, and the stimulation of glutamate allows for the excitatory reactions. Now, because of the reduction of GABA, you have an increase of sodium and calcium, and because of the glutamate, you have changes within the brain-derived neuron factor, which promotes survival of neurons. However, this does need to be studied more because it suggests that there is causes of modulation in the central nervous system. Now, the regulated transmitters are highlighted through the pharmaceutical studies, where they found DMO suspended the after effects of TDCS and other psychotropic drugs, altered the effects of TDCS, which is how they were able to determine 3 key transmitters that are directly altered by TDCS: dopamine, serotonin, and acetylcholine. Now, this then is preceded by a cascade effect that then is expressed through phosphorylation, and then finally an increase of protein synthesis, what strengthens the neuroplasticity of cells. This here is the model. Feel free to pause it to get a deeper understanding of transcranial direct current stimulation after adrenal stimulation. Now, in 2008, a researcher, Grime, found a theory of hypofunctionality between the left and right dorsolateral prefrontal cortex. Now, what this means is there is a neuronal imbalance of activity between the left and right dorsolateral prefrontal cortex. The left experiences hypoactivity and the right experiences hyperactivity. Now, the difference between hypo and hyper is that the hypo is reduced or decreased activity, whereas the hyper is increased or spontaneous activity. And now this has been shown to be improved after stimulation, and it also has been tested pre and post treatment with the BDI, the MARS, and the HAMD depression rating scales, as they all show decreases in symptoms. Now, this is also shown in another study done 8 years after the Grimes study, so it has validity there. This slide shows the data retrieved from the 2008 study done by Grimes and colleagues. On the left-hand side, you'll see the clinical characteristics of the depressed and healthy subjects. And on the right-hand side, you'll see the measured data of hypofunctionality and hypofunctionality within the left and right dorsolateral prefrontal cortex. If you move your eyes down to the graphs of C and D, you'll see there's two different colors. The dark gray is the healthy patients, and the light gray is the major depressive patients. And within the top one shows the left dorsolateral prefrontal cortex, which highlights, you can see that between the healthy and patients, you can identify that there is hypofunctionality within the left dorsolateral prefrontal cortex, and then on the right hand side, with D, you can understand there is hyperfunctionality measured in the right dorsolateral prefrontal cortex. Now, this table shows the response rate and the remission rate followed by stimulation. The first 3 studies place the anode on the F3 and the cathode on the F4, and the next 3 studies place the cathode on the supraorbital lobe, which has been found to have more of an increase within reactions. Now, please feel free to pause it to grab a clear understanding of how much better the reactions are. Now, limitations in future research. Limited knowledge surrounding the interactions made between the different systems within the brain and neuronal networks. Experimental variability. That means the intensity, duration, and placement of cathodes can all be altered to have different results, and individual variability highlights that every brain is different, and so by placing one cathode or anode in the same place as on another person, you might get different results. And future research, there is a relation between the Alzheimer's and reduction of glutamate or GABA, and so maybe more research into how to produce glutamate or GABA will prolong the onset of Alzheimer's and the improvement of cognition after stroke. Now, it's highlighted that the stroke probably has the best cognitive improvement after TDCS, and it also is noted that those suffering from treatment-resistant depression do not have any significant improvement after TDCS, so maybe research into treating how TDCS can improve treatment-resistant depression in future. And then here is my references, and have a good afternoon.